Hi, I'm Marc Rothenberg at Cincinnati Children's Hospital in the United States, and I'm very grateful to Sarah Gray for inviting me to present to you for your National Eosinophil Awareness Week. Thank you so much for all you do down under. Today, I'd like to take you through our research, primarily focused on eosinophilic esophagitis, which has been the subject of my research. I want to start by updating you about the eosinophil. The cell was described in 1879 by Paul Ehrlich, and it was identified and named because of its intense staining with the acidic dye eosinophil. Hence, eosinophil for attraction or love of the dye eosin. The cell was described as originating from the bone marrow, and it was a circulating granulocyte, counting for only a small fraction of the total peripheral blood cells, 1%. However, it was later appreciated that the cell was also a tissue-dwelling cell, and the primary location for eosinophils is actually in the GI tract at baseline, although the esophagus is devoid of eosinophils. It's also notable that, unlike other white blood cells, eosinophils do not divide. They do not proliferate. They merely accumulate in the tissue in response to inflammatory triggers or during homeostasis. They can function as pro-inflammatory cells in select diseases. In other cases, their function remains unclear. Here in this picture, we can see an eosinophil, a beautiful cell with its eosinophil granules and the blue part being the nucleus. Eosinophils respond to a variety of different triggers. On one extreme, allergens, the most common cause of eosinophilia, an increase in eosinophils, but also other triggers, including infections such as parasites, viral infections, and tissue injury. We now understand that these are pleiotropic cells capable of making a variety of different mediators and participating in a variety of different responses that are critical for a number of different communications with the cells and tissues in the body, such as the nervous system. Now, diseases associated with eosinophils are typically called eosinophilic diseases. Eosinophilia is the increase in eosinophils. Now, this can occur in the peripheral blood or the tissues or both. Let me give you some examples. In infectious diseases, we can have an increase in eosinophils in the blood and sometimes in the tissue. This would be an example being an invasive helminth or a parasite. In respiratory diseases, eosinophils are typically absent in the blood, but they can be present in elevated numbers, but they are by definition present at increased levels in the respiratory tract, such as in the case of eosinophilic pneumonitis or asthma. In the GI tract, we can have peripheral blood eosinophilia on occasions, typically not an EoE, but it is a disease characterized profound eosinophilia in the tissue, particularly in the regions associated with the inflammation causing the symptoms. That includes inflammatory bowel disease, eosinophilic gastrointestinal diseases such as EoE, and allergic colitis. And the list goes on, and I provide this along with the reference to give you an idea for the spectrum of diseases associated with eosinophilia. Now, my lab has been focused on understanding eosinophilic diseases. We take multiple approaches. On one end, we have the fundamental research operation in my lab, which is focused on traditional approaches of basic science, including signal transduction and molecular analysis. We have a genetics arm in my lab, which looks for the heritability of eosinophilic diseases, trying to scan the whole genome for susceptibility regions and understand how diseases are inherited. We also have a component of my lab which is focused on translational medicine, meaning that we can model eosinophilic diseases in the test tube, in vitro, or in preclinical models, such as animal models or ex vivo modeling of the disease as well with human tissue. Now, this is a very dynamic process where we could have one hand a genetic lead, a hit through a genome-wide analysis. Then we go in the lab to see if there's any functionality If there is, we can go in and test that function in some of the translational medicine systems. Now, if these three areas converge on positive data, then we go in and test these theories in patients. And we have been able to move from fundamental discovery all the way through clinical studies, aiming to improve life and health of patients with eosinophilic diseases, especially EoE. I happen to be directing the Consortium of Eosinophilic GI disease researchers in the US. Our mission is to improve the lives of individuals with eosinophilic gastrointestinal diseases through innovative research, clinical expertise, education via collaboration between scientists, healthcare providers, patients, and professional organizations. My partner in this leader is led by Dr. Glenn Ferruta. I'd encourage you to visit the CEGIR website, where you can get a lot of information about eosinophilic diseases and learn about what we're doing. The consortium is focused in the US. It has 18 national sites. It has 100 members that are active in the disease. We're supported by various patient advocacy groups, as well, importantly, as primary funding from the National Institute of Health. There's a number of different components. It's a complex operational management system, but importantly, we have great amount of expertise, people working together with the aim of better understanding and treating eosinophilic diseases. Patient advocacy groups are central to our mission. Patients and this organized groups such as CURED and ABFED help us with trial design and recruitment. It helps us understand and disseminate the research findings, educates other medical professionals and the public about the findings, influences policy, funding decisions, interacts with the Rare Disease Consortium network at the NIH, and helps us shape the research agenda so that we make sure that the work of CEGIR is focused on meaningful research projects that are affecting and important to patients. A number of years ago, 22, I described the whole family of eosinophilic GI disease researchers and coined the acronym EADs. Importantly, at that time, we classified EADs as being intermediate spectrum between pure IgE-mediated disease, such as food anaphylaxis, and a non-IgE-mediated immune disease, such as celiac disease. The different types of EADs were defined based on the involvement of the GI tract, ranging from the stomach to the esophagus, multiple segments, which we now call multi-segment eosinophilic GI disease, as well as enteritis, the small intestine, and the large intestine. Recently, my colleagues have been looking across the whole U.S. Medical records, 300 million electronic medical records for the prevalence and incidence of these diseases. And we can see in this data set that the prevalence is rising. The numbers in the US are quite substantial, up to 500,000 patients based on coding. The incidence also continues to rise, at about 1 in 5,000 new patients per year. We can also see that this disease runs with other atopic diseases, other allergies, and depending upon whether or not you have these allergic diseases, it will greatly influence your odds ratios of developing EoE. If you have all forms of allergic disease, if you happen to be that misfortunate, such as suffer from asthma, allergic rhinitis, eczema, and food anaphylaxis, your risk for EoE is as high as 24-fold, which is a huge number. In the US, we now understand the distribution of this, and it's very interesting to see that the prevalence is higher in the northern states, and this is consistent with a environmental etiology, and I won't go into that today, but there is a strong environmental component that works together with the genetic elements, which I'll come back to. A number of years ago, as we began to describe EoE, we didn't understand what was its cause. So we conducted some seminal experiments in mice, in this case proving that an allergen exposure in a mouse elicited esophageal eosinophilia. So if we just gave the antigen orally or intragastrically, put it in the stomach, we wouldn't develop a break of tolerance. But if we applied it intratracheally or intranasally to an anesthetized animal so that they aspirated it into the lungs but also swallowed it at the same time, they developed a profound eosinophil accumulation in the esophagus, demonstrating a couple of points, but one, and most importantly, is that EoE in mice and humans is triggered by allergens. So it's not a reflux or acid-associated disease, but it's an allergic disease. And we know what allergens are causing EoE because my colleagues and I in CEGIR conducted a randomized clinical trial where we avoided the 6 most common foods, called the 6-food elimination diet, or only milk. And what we found in this study was quite surprising, that milk elimination alone was equally as effective as avoiding 6 common foods. Now, the good news is that diet works. The tough news is that it only worked in this particular study, which was a controlled, randomized, multi-site study, which is a really strong study designed for this disease, was that only 35% of the patients responded. So my colleagues and I have concluded that milk elimination is a reasonable first approach for treating EoE, and that some patients do respond, and we have some biomarkers to predict response, but that many patients don't. But this is a reasonable first approach. Now, we do know that eosinophils are not the primary driver of the disease. So we are looking for mechanisms beyond eosinophils. We now understand that EoE is a hereditary disease involving complex genetic environmental factors, and this was demonstrated in our twin studies. This was an international registry that we set up. We asked for patients around the world to send us information about their EoE if they were twins. And we also got their DNA samples. Interestingly, we had a number of people from Australia that signed up for this registry. And we found some very interesting findings. One was that indeed there is a genetic component because monozygotic twins had a 40% concordance rate. But that also shows that even if you're genetically identical, monozygotic twins, you still have only a 40% chance of developing the disease, indicating that genetics is important, but also other factors, particularly environment. Now, we were most struck by the finding that dizygotic twins, which share the same amount of DNA as regular siblings, shown here, had a nearly tenfold increased risk for the disease, indicating that it was the early life environment that was leading to the susceptibility for the disease. That early life environment is the one that's shared in utero and also similar in the first year of life as the babies are in the cradle. And we now know through other work that early life factors such as C-sections and antibiotic exposure and even proton pump inhibitor usage are major factors in early life. Now, in terms of the genetic components, my colleagues and I have deciphered that by conducting what we call a genome-wide association study. And this is data that we call a meta-analysis, which is putting together a lot of studies where genetics have been used. And this looks at a couple of thousand people with EoE and many times more controls without EoE. And we're looking here across the whole human genome, and we're identifying regions that lead to susceptibility for the disease. The y-axis shows the statistical significance, and this dashed line shows the regions in the human genome that lead to susceptibility. These are the chromosomal positions. Each of these dots is a different genetic variant. And one can see here is that we have peaks. This is actually called the Manhattan plot because we have all these peaks here, which are like buildings. And then we have skyscrapers that are crossing this line here. And these are the regions of genetic susceptibility. Now we know what's sitting down in these regions in the genome. And today I'll just draw your attention to this region here, because this is the strongest region that leads to susceptibility. It's at chromosome 5, Q22. And sitting right there is a gene called TSLP. This is particularly important because the TSLP gene codes for thymic stromal lymphopoietin. That's a long word, but the important point is that it is a cytokine. It's one that's critically involved in the development of allergies. It's one that we can target with an antibody that's in clinical usage now for asthma and is being studied for EoE. And this is testing whether or not the TSLP may mediate the disease. In this case, we're looking at mice that are overexpressing the TSLP gene. And what happens is they overproduce the TSLP protein, particularly in the esophagus. And what we found was that this was very sufficient at eliciting EoE-like changes in mice. The anti-MBP is a stain for eosinophils. This is the WT or the wild type mouse that's non-transgenic compared to the TSLP transgenic, the mice that overexpress the TSLP. And one can see that the staining goes up and that all these dots here are eosinophils. And this is looking under the microscope and staining the tissue. And we can see the mouse esophagus here. This is the lumen where the food goes through. This is the epithelium. This is the subepithelial area. And one can see that the architecture of the esophagus is greatly altered in the mice that have overexpression of the TSLP. Not only is there a thickening of the epithelium like we see in patients, but there's disorganization of the epithelial layer. There's an epithelial expansion. There's also hyperplasia, which means that there's increased proliferation rate of the epithelium, and we can quantitate that here, which we can see substantial differences that are statistically different between the transgenic mice and the wild type control. And this indicates that overexpression of TSLP, which occurs in the human disease, EoE, is sufficient to induce the EoE, greatly supporting the genetic susceptibility in this region, being mediated by the TSLP protein. Now, in terms of understanding the disease beyond eosinophils, it's important to note that the environmental factors interact with the genetics. And we do think that this is mediated by the microbiome, which are the bacteria that normally reside within our body. What we've shown in the past is that the esophagus harbors its own unique bacteria. It's called flora. And that this flora is dominated by a particular bacteria called Firmicutes, which includes the species Clostridium. What's notable is that the microbiome is required for the maturation of the esophageal epithelium. That the microbiome can be disordered by treating the mice with antibiotics. And then if we take the microbiome that's associated with dysbiosis, that will lead to an elevation of IgE and a predisposition to EoE in mice. Also, we have demonstrated that we can transfer the esophageal microbiome by a fecal transplant, basically meaning a stool transfer in mice is sufficient to modify the esophageal microbiome. These are fundamental findings indicating that the microbiome plays a role in the esophagus and that it is manipulatable, and it may explain the environmental factors that are influenced by the early life exposures that lead to EoE susceptibility. Thinking beyond eosinophils, it's important to note that the epithelium of the esophagus has a contributory role for the disease. My colleagues and I have spent many years working on understanding this complex tissue. We now know that the esophagus isn't just simply a conduit to pass food from the mouth to the stomach, but it's actively involved in homeostasis and protection of the host, a person, from the foreign substance, the food, from responding to the biophysical challenges such as rough surfaces, hot temperatures, spices, and other things, and that there are strong barrier proteins that hold this together so that the esophagus prevents food from getting absorbed. It's a barrier. Now, in the disease state, we know through a number of studies that some of the genes that we've identified, some of the triggers that cause the disease, weaken the barrier. You can also have genetic forms of EoE that's associated with loss of function of some of these proteins, in particular the barrier genes The calpain 14 is a common variant. It's also present. Its gene is part of the GWAS studies that we conducted in chromosome 2. And this TSLP, as I said, is constitutively expressed by the esophagus. And there's a number of other interesting features that allow the esophagus to normally respond to different events, traumatic events. But importantly, when there's a perturbation in the system, we think that sets up the individual for the development and propagation of the EoE inflammatory response. We also now understand beyond eosinophils that the esophagus is equipped with nerves, sensory nerves that run from the spinal cord as well as the vagus nerve into the esophagus and set up a whole network that allows the esophagus to respond to different triggers, for the patient to feel different sensations. But this is not just a sensory arm, but it's actively communicating with the immune system through a process called neuroimmune networking. This is an image that we now have of the esophageal nerves. And in white, we see a certain type of nerve which forms a network, a web that surrounds the esophagus. The blue is the esophageal epithelium, and you can see the lumen, which is where the food runs, is over here. And right below the epithelium is red staining, which is sensory nerves that run longitudinally along the esophagus, providing multiple ways in which the nervous system is sensing, but also interacting with some of the immune cells, including the eosinophils and mast cells. This is an image also of the esophageal nerve staining, and we're basically looking at this in 3 dimensions, and we're rotating the image, and one can see the beautiful staining that we now have. But really what I'm trying to show you is the actual patterning. This is a different way of looking at this. It's the lumen of the esophagus, and then you see the epithelium, and then you can see surrounding is the network of nerves, and the red dots are the actual sensory nerves that run in juxtaposition to the epithelium. And you can see it's sitting right underneath the epithelium, and it turns out that they send projections into the epithelium. And this, we believe, is very important for interacting with the epithelium, but also potentially triggering and eliciting some of the inflammatory responses. We have shown that there's an increased innervation in EoE, there's an increased number of nerves. There is particular signaling pathway related to dupilumab with the alpha receptor alpha on the nerves, and then knocking this receptor out or deleting it or interfering with it attenuates inflammation when the alpha receptor alpha is specifically interfered with on the nerve cells. So we think the dorsal root ganglia directly responds to the type 2 cytokines. And the human data that we have, which is not shown here, substantiates that these findings are operational in patients. And we also have evidence that dupilumab reverses some of the hyperinnervation that we see in the disease. So this is a new area, a line of research that we have, particularly focused on neuroimmunology, which I think is not only important for listening disease, but may explain some of the refractory symptoms like pain that patients get. And this is a model of what we now think is happening, which is that the local tissue is responding to the allergen, triggering the IL-4 and IL-13. This causes an activation of the nerves through the dorsal root ganglia. This triggers a response to the central nervous system, but importantly also triggers in a feedback loop in a bidirectional manner. It modifies the inflammatory response in the esophagus. So mechanisms beyond eosinophils include an integrated mechanism that involves the genes, the environment, and multiple cellular and cytokine signaling pathways, which is shown in this image here. This is our working model of EoE pathogenesis. We know that there are a number of components of the epithelium of the esophagus that are critical, that there's a barrier impairment in a disease, that there are particular proteases, which are basically enzymes that repair the tissue that's dysregulated, particularly because some of the anti-proteases like Sphinx are deficient in EoE. This triggers overproduction of the TSLP. TSLP targets innate immune cells, particularly dendritic cells. and also mast cells and activates them so that they can trigger innate and adaptive immunity, particularly the food reactive Th2 cells that then generate IL-13, which is a very critical cytokine that's upstream and it's the target of dupilumab. What IL-13 does, it's a potent inducer of transcriptional. It activates eotaxin 3, which is an eosinophil chemotactic factor causing eosinophils to infiltrate. It also activates the calpain 14 transcriptional machinery so that this is overproduced and this affects the barrier function or the homeostatic dysregulation that takes place. We know through rare genetic mutations in humans that you can have loss of function of the sphinx proteins or the desmosomes, which are the proteins that hold the epithelium together that lead to EoE. So this is our working model of the disease, and we have multiple lines of independent evidence that this is operational and targetable with different drugs. And we do think that reversing this and re-educating the immune system will be very critical for eliciting long-term remission and cure of the disease. These are some of the pathways that I've described today. The complex but well now understood pathways that lead to tissue remodeling, loss of barrier function, eosinophil inflammation, nociception, which is the sensory arm triggered by various stimulants that modify the inflammation, but also patient behavior. We now have capacity to block a lot of these elements, whether it be IL-5 blockers, eosinophil depleting antibodies, IL-4 and IL-13 inhibitors, TSLP inhibitors that are in late-stage development for EoE, IL-33, another molecule related to TSLP that we can have antibodies against, mast cell depleting drugs that are very important, and inhibitors of enzymes like JAK inhibitors, JAK kinase inhibitors that are also available to treat patients, and we're currently pursuing them, as well as modification of the protease-antiprotease balance, such as increasing the level of the antiproteases like Sphinx 7. I want to close by saying that besides developing anti-inflammatory drugs, I'm now heavily engaged in trying to think about the future for the field. And one of the areas that we're very focused in on is to develop mRNA lipid nanoparticle immunotherapeutics. This work started during the COVID pandemic when my colleague and friend Drew Weissman developed the technology for mRNA vaccination, subsequently won the Nobel Prize. Instead of putting the spike protein from the COVID virus into the mRNA vaccine, I asked Drew if we could put food allergens in there. And lo and behold, he said yes, it was a good idea. And he constructed the mRNA vaccines. And instead of having the spike protein, had the allergens. And what we found is that this modified the immune response dramatically. It produced a change in the T cell. It produced an increase in antibody levels that were not of the IgE type and shut down the allergic response. And we worked on this subsequently since 2001, applying this new technology to understand how we can re-educate and rearm the immune system to reverse, if not prevent, allergic diseases with a strong focus on EGID. This is data showing you that this is a very potent way of re-educating the immune system. This is a healthy mouse. This is a mouse with allergic disease that have gotten a control vaccine. And these are mice that have a vaccine. And you can see they develop antibodies against the allergen. And it's not the IgE that they develop, but an IgG, which we think is very critical for potentially neutralizing the allergen. We can shut down the cells that are making the type 2 cytokines like the IL-5 and IL-13. And indeed, the mRNA vaccine is very effective at lowering the level of the Th2 cell. We can also improve different aspects, including reducing eosinophilia, in this case in the lungs, dramatic decline in eosinophil levels with the mRNA vaccine. We think that this is a platform technology, and we're looking forward to applying this to all forms of allergy, starting with EGID, allergic asthma, anaphylaxis, and food allergy. More to come in the future, but I wanted to share this exciting new development with you. I want to thank all the groups that have been contributing to this, the researchers, the allergists, the GI specialists that I'm fortunate to work with, pathologists, research coordinators, the research lab, others, geneticists. Thank the organizers of this meeting and the NIH in particular for the support to allow this work to happen. Thank you very much for your attention today.